complement system. And also if you've got time, Matthew, an update for the Swedish conference that was held in Sweden, obviously. Late June, July?
Yeah, that's right, it was June. Yeah, we will have time. Yeah.
Which Matthew will explain why that came about, and we'll give you a full update.
So, thanks very much, Andy. It's a real pleasure to be here. And what I thought I'd do is give you an overview of complement and then the update, because I know some of you have heard the sort of previous iteration. Complement's central to C3G, and C3 is a complement protein, so you know that's the clue in the pathogenesis. So I work down in London, and we work in a renal unit. I provide the complement expertise, and in association with a renal physician called Dr. Tom Cairns and a superb nurse called Sister Nicola McKenna, we see patients with C3G, MPGN and DDD in addition to other disorders. So let's ask the question, I think, what is complement? Let's think what it should be doing, and it's part of the immune system, as you've heard from Kevin. It's a complex protein network, and the details aren't really that important. They're not important, particularly in some aspects, to even researchers in C3G. It's a network that's there to fight against pathogens, but not just any pathogen. It's predominantly bacterial infection that's important in terms of complement. So this network destroys bacteria. And I'm going to describe the mechanisms, how it does it, and describe it as firepower, because there's different approaches to how complement achieves this target. So let's think about the firepower, because when that goes wrong, that's what's going on in the kidney. So if we understand that, we can understand how the drugs that influence complement might make things better. So the firepower can kill the target directly. It can mark it for destruction, recruiting those inflammatory cells that Kevin talked about, and it can call for help along similar lines. So here's a schematic just to show you this, you know, conceptually. Killing the target directly is probably the best known function for complement. It's the one that medical students, doctors will remember and talk about. And it does this by forming this complex that is essentially like a toilet roll, really, that goes into the membrane of a cell and allows things to leak out of the cell, and the cell will explode. And we don't call it a toilet roll, that wouldn't sound very sophisticated. So like we do in medicine and science, we call it something a bit more, we put the jargon in, which is a membrane attack complex. And it allows, as I said, things to leak out of the cell. So this is what will happen to a bacteria. And this shouldn't happen to host cells, but can do if we get the target discrimination wrong. And the bacteria become leaky and explode. I won't spend so long on these slides because they're on the YouTube, and you can go back and look at them, but I'll just illustrate the principle. And we can actually see this. We can take pictures. You're probably used to seeing these electron micrographs of your kidney basement membrane. This electron micrograph, same sort of thing, very, very detailed microscopy that shows these holes on a surface. So this is a very interesting and powerful mechanism of attack by complement. The other thing is we can mark bacteria. We can help to differentiate host and non-host by labeling proteins on the bacteria that tell those fighter pilots that Kevin talked about that that's the correct target to identify. And that's something, again, that is sometimes known as opsonization. And the phagocytes, these inflammatory cells, etc., come in and target the bacteria. And then there are help signals as well. Complement sends out signals that attract these cells to the site of injury. So all of this makes intuitive sense, doesn't it? All of it is got the single aim of damaging that target optimally. And that's shown there. So that's the firepower. And when we think back, well, how within this protein network, I don't want to go through the details of the protein network, but let's pull out two of the proteins, C3 and C5. They're really the key components that, if you like, unleash this firepower. All right, and we need to know a bit about that, don't we, to understand C3G, because it's in the name. So here's, I'm just trying to get here we are. Here's C3. This is what we see deposited in the kidney, and that's why we call it C3 glomerulonephritis. When we look in a lot of samples, we see C5 as well, so we could call it C5 glomerulonephritis, or if you like, C3 and C5 glomerulonephritis. Okay, but you get the point. So the complement proteins are present in the kidney, and therefore the worry is that these the consequence is the firepower is also present within the kidney. All right, and that's going to cause kidney inflammation. So we think of ways of switching that off, and we'll talk about C3, switching off C3, what we call C3 inhibitors, and switching off C5, which is the famous Eculizumab that I'm sure everyone has heard about. Okay, so again, we have to discriminate, don't we? We want to destroy the bacteria. We certainly don't want to do this in the kidney. So this goes back to this self, non-self paradigm that comes up all the time in immunology. It doesn't matter what you're talking about. We're focusing on complement now. So we need to think about how complement is switched on. Well, Kevin's talked about antibodies. That's a classic way that complement will be switched on, and antibodies are very, very specific. Some structures on bacteria are only present on bacteria, so you don't need antibodies to recognize bacteria. If you can recognize those structures, and they're, for instance, sugars on bacteria, and there's a whole system where complement is switched on by recognizing sugars that are only present on bacteria. And of course bacteria get clever, so they modify the sugars to stop that pathway from switching on. So all of this can be subverted by different bugs, and that presents, you know, danger to us. And then we have the pathway that really is the mischievous one for the kidney in this condition, and this is the alternative pathway. This is like attack everything, don't worry about discrimination, attack everything and rely on the fact that your own cells are able to protect themselves. Okay, you know, and that might seem reasonable provided your host cells can protect themselves. So that's shown there, attack everything, including one of us, and again, if you don't have appropriate ways of switching off complement on the surface, then we're going to get damaged by our own complement system. So how is it switched off then? Well, it's switched off by just that there's a protein network that switches it on. There's a whole protein network that switches it off. And this is a very complex system, as you can imagine, because again, why is it complex? Because the bugs are complex. When the bugs start to evade different on switches and off switches, and over evolution, we start to develop ways to counter that. So we call these complement regulators, and some of them you know will be familiar to you because they may be, you know, you may have heard about the genetics. If you heard about factor H, factor I, CD46, and so on, these are regulators, and we're looking for problems in those regulators, those off switches, when we're doing the genetic screening. And there's factor H and factor I. So when the proteins are present on those cells, we don't get any damage, and we have a sort of steady state, normal, healthy state. Now, the implication of all this is, well, okay, if you have a complement-mediated disease, the strategy is simple, isn't it? Let's just switch off that firepower in some way. Take away C3, take away C5, take away both. And before we do that, we have to think, well, what are the potential consequences of that? Is there anything harmful? Well, I've said to you that complement is important in infection, so would you be more prone to infection if we switch the firepower off? And we can take some lessons from people who have been studied, who are born without C3, who are without C5, and there's lots of other examples. And what we see in those individuals is a susceptibility to infection, particularly bacterial infection. The good news is it tends to not be a problem as you get into adulthood, because you have, although you're missing this firepower, you have, and this should come up if I press this now, if we take this away, you're missing all this. If you have C3 and C5 deficiency, you have these things. You have antibodies. You've still got those missiles coming out of the fighters, and they can compensate, and we're reasonably confident. But therefore, if you took away C3 with some sort of antibody, you know, not Eculizumab, that's C5, but if you took away C3, we think people would be okay. This is something that people would tolerate. There are caveats, of course. One of the dangerous bacterial infections that C5 is very important at fighting is meningitis. And that's why when people go on to Eculizumab, we give treatment to combat that. Okay, but we know that if you're vaccinated and you're on antibiotics, then that's a safe treatment. The risk-benefit is in favour of inhibiting C5, and I think the same will be true of C3. Okay, so let's go to what happens when there's too much firepower, and we're going to focus on C3 glomerulopathy. Okay, and we're including DDD and certain types of MPGN in that. There's an imbalance the off and on switches are dysregulated. Right? And this is what we're looking at when we look at the genes. We're looking for changes that result in damage to the off switches, or actually not damage to the on switches, but make them super functional. Okay? So now that they're doing their job too well, so even though the off switches are good, the on switches are too powerful, if you like. And that's at the genetic level, and also then in the blood circulating. And Kevin's shown you one acquired factor that interferes with this system, and that's these autoantibodies that antagonize and block the normal action of factor H. When we're looking at the genes, somebody asked about an exome and a genome. I'll just digress. The genome is huge. Okay, if you add up all the units, which we call nucleotides, in one of us, there's 3.2 billion. So imagine that's a long sequence to look at. So what we concentrate on, by and large, in most genetic studies, not all but most, is just the regions where the genes make proteins. Okay, that's called an encoding region. And that's referred to as the exome. And the good news about the exome compared to the genome is the exome is about 3% of the size of the genome. So if you're going to look at the exome, it's a lot more simpler, the data set is smaller, and arguably is the place to look first, because this is where the proteins are encoded. Now we've got 24,000 genes in a genome. And if you send your DNA off to look at the genetic factors associated with C3 glomerulopathy, we look at factor H, factor I, CD46, C3, and factor B. So 5 genes, sometimes a few others. So that's when you get a genetic screen back that comes back as normal, we're saying at 5 or 6 genes out of 24,000 are normal. Okay? So what we hope, and Sally presented very nicely on the link with the Cambridge bioresource, is by looking at all the rest, maybe we'll find previously uncharacterized causes of this condition. Let's not make the assumption that there's no genetic causes, let's look at all the rest. Okay, but you can see that's a big task, 24,000 genes. Well, we've done 6. Okay. Right. So if we have too much firepower, we have this imbalance, we get this, let's say MPGN, and all the other terminology. And we're interested in the firepower going off inappropriately in the kidney. There's a lot of research going off studying the firepower going off inappropriately in other organs, in the eye, for example, okay, and on blood cells and various others. And that's good, because that has the advantage for kidney and complement problems, is that drug companies, whatever they find, perhaps to do with eye disease, will be repurposed for treating individuals with a problem in the kidney. So a lot of interest in complement by companies. So this is C3. This green picture here is it's usually always shown in green. Is C3 in the kidney, and of course that shouldn't be there. There is no antibodies there. There are no bacteria. There's no bacterial sugars. That's inappropriate, and that tells us usually that there's a problem with that alternative pathway. And this is what happens. And again, these are slides that are on the YouTube and on the internet sites that you can look at. And it's just showing you conceptually what will happen. So, too much firepower in the kidney. Now, I put this slide in just to think a little bit about treatment, and just emphasize that one thing that strikes us about C3G, which is a different to, say, atypical HUS, that again some of you may know, is it's very variable, this condition. Okay, so I imagine all the stories that you have amongst yourselves, there'll be a lot of common things, but there'll be a lot of differences. And this just shows you biopsies from patients with C3G, and this is dense deposit disease. This is another subgroup over here. And I don't need to go through the detail here, but you can tell these pictures look different, don't they? So there's different types of inflammation going on in the kidney. All right, all of them share the feature that there's C3 in the kidney, but the inflammation is very different. So what I propose is, I don't think we should think, is Eculizumab a good treatment for C3G? Is C3 a good treatment for C3G? We should think, well, at what stage in the illness might those treatments be useful, and for how long? Okay, it's not just necessary. I don't think it's going to be one treatment. It's going to be maybe manipulating different aspects of complement for different periods at different stages in the illness. So what have we got to control this firepower? At the moment we've only got an antibody. It's an antibody that's been designed not to activate complement. Okay, it does the opposite. It binds to C5 and switches off those two elements of the firepower. All right, those two elements of the firepower are very important in fighting meningitis, so we have to remember that and protect patients on this treatment from meningitis. But if the disease is being caused by C5 activation, then this should be a very, very good treatment. If the disease is caused by C3 activation, or there's a significant part of it that's caused by C3 activation, this won't be enough, will it? Because C3 carries on activating. What if you took away C3? Well, that has the advantage now that because C5 is downstream, as you can see on the picture, you take away both the C3 mediated damage and the C5 mediated damage. So you might think that this would be a more powerful approach to take away C3. Okay, I'm going to switch now to the update from the C3G meeting, which was in June. I was actually struggling to remember. There's been so many meetings this year. June the 26th, and it was in Sweden. The reason it was in Sweden was because that was where a big international meeting on complement was taking place. So we took that opportunity. We had a lot of complement biologists who do lots of things other than the kidney. We feel sorry for them. They're not where the most important organ is, of course, but anyway, they're there. And we had this just before the meeting. So we had a lot of expertise in the audience. And we wanted to build on really our last synthesis of the literature, which was this report, which is online, and the open is important. It says open on it, doesn't it? It means that anybody can get it anywhere in the world free of charge. And that was the sort of consensus, the expert group view of where we are with C3G at that time, 2013. This was 2 years on, essentially. And it was co-organised by myself and Richard Smith, and here's a picture of Richard. This is not in Sweden, this is in Hingston. And hands up if you know Richard Smith, or you know of him. One of the most remarkable individuals I think I've ever met. I mean, a very, very dynamic, kind and caring person, and somebody who's really making big inroads, I think, into the global awareness of this condition. And I just thought I'd say that because I think a remarkable individual. So we formed this program, and how do we, I'll just show you excerpts from the program so you can see how we thought about it. Well, we wanted to know more about the renal pathology. Is all this subdivision into all these different names and different types, is that going to be clinically meaningful? It might be interesting for genetics, but will it matter for treatment? I gave the example at lunchtime. If you have high blood pressure, there's loads of causes of high blood pressure. Treatment's generally the same. Okay? We need to understand whether our subdivisions are going to be meaningful for treatment. All right? So that was one aspect. Another was the clinical phenotype and assessment. What should we be doing in patients in addition to the genes we've mentioned, the factor H autoantibodies? Kevin's doing more and more clever autoantibody tests. Should we be sending samples to Kevin to get complete analysis in every patient, for example? What do we know about the driver's disease? There's updates on genetics, acquired factors, autoantibodies, and so on. And then there was this, which is perhaps what you'd be most interested in, is focusing on what we do with treatment strategies. We talked a lot about Eculizumab, and I'll show you some slides on Eculizumab. And we talked mainly offline about emerging complement therapeutics. I'm contacted more in the last 18 months by companies developing complement therapeutics than I've ever been in. And I've been in the game for like 20 years or so. So we really are coming at a time where these companies are starting to think seriously about moving into trials. All right, and that's good news. I mean, it's good news if they're developing products, but not so good news if I'm telling you maybe that's 5-10 years downstream. Really, the discussions we're having is how would we do the best trial to see if this complement inhibitor works. The problem when you deal with companies, and it's not a problem that you have to protect their intellectual property, so you can't talk freely about specifics, but the general approach everyone knows. And the message I want to give you is there is a lot of intense interest at the clinical trial level. Okay, So here are some of the updates, and we've talked a bit about this. I've talked about really the renal biopsy, how meaningful are all these sub-classifications, all right? That's one point from that, and you don't need to, the detail on these slides are not important. The clinical assessment and phenotype really was about trying to stratify patients, trying to look about who are stable, who are going off acutely, etc., and thinking about how we would position the different types of patients at different stages in their illnesses into a trial. Okay, and that's not a straightforward question. And then we talked about the treatment strategy. I've talked about emerging therapeutics, but the other question that came up and was discussed a lot was how to utilise Eculizumab. So obviously Eculizumab is licensed for conditions, it's not licensed for this condition, so it's out there being used. How do we utilise it in C3G? And what evidence have we got? Well, we have the literature, 27 patients you can see in front of you with mixture of diagnosis, DDD, C3GN, some treated in native kidney, some in disease that's recurred in the transplant. And of course in these meetings people will talk to you about cases that are not published as well. So there's certainly more than 27. But that's the current state in the literature. And when you look at the literature, you don't see a clear message, you don't see a message that nobody responds or that everybody responds. You see that some people respond, and some of the response is very, very meaningful. And I've just put some examples here, allowing some patients to come off dialysis, okay? Making nephrotic syndrome better, allowing people to reduce drugs. Okay, so there's clear benefit in some individuals who've received this drug. But in some cases, and that's the bottom part of the slide, you can see there is no response. So the question is to understand why is that, right? And how do we stratify those patients? And it's really thinking along these sort of terms, is treating the disease in the native kidney, treating it in the transplant kidney, and of course when you have a transplant, is there any role for this drug in stopping the disease coming back, should you think that is likely? Earlier this year, and again, you know the background to all this, but NICE have produced this evidence summary on the second point. Okay, what is the evidence? Really focusing on that specifically. What is the evidence for Eculizumab in preventing disease coming back in the kidney? Okay, and they'll be looking at other aspects of those questions in due course. I mean, the bottom line, as you might expect, is because of that, some response, some do not respond in disease, not in prevention, in disease, is that we need more evidence to understand how to use this drug. And we talked a lot about how we might try to achieve that in the meeting. You all know that the cost of Eculizumab, and that does put up there's no point pretending otherwise, that does put up a serious barrier to moving forward to answer some of these questions. This is just really towards the end now, is just to show you my point about different treatments at different stages. So this is just taken from one of my friend and colleagues' paper, Fadi Fakuri, who's a nephrologist in Nantes. And this is a patient with C3 glomerulopathy. And when you look at the renal biopsy, you can see, I'm anxious as I touch this, here. You can see C3 in the kidney here, and you can see C5 in the kidney. The more dark it is, the more of that complement protein there is. And this is in months here, 8 months difference, and then 9 month difference. And you can see here that look at the amount of C5 at this time point. So if you took these 3 time points and said, when would you most want to give an anti-C5 antibody, you'd choose the middle one, wouldn't you? When there's evidence of C5, in copious amounts of C5 in the kidney. And of course that's what they're showing here. This patient did get Eculizumab and got better. There was an improvement. And I think what this is telling us is that biopsy is saying at that point, C5 firepower was causing a lot of the damage. Maybe if you gave it here in the first column, the amount of C5 contributing to whatever the patient was complaining about would be relatively less, and the benefit wouldn't be as marked, if at all. But that wouldn't be to say Eculizumab has no role, would it? So that's where we are now in 2015. We are looking. A lot of companies have agents that will switch off C3, not directly, but usually coming in by other protein networks, but it'll culminate in the same effect, switching off C3. And I really believe that trials will start reasonably soon, and I'll stop there. And just thank again Andy and all of you in the support group for inviting me. Thank you.
Anyone got any questions?
I don't know if it's really relevant to you, but I just wondered, do they still think that DDD affects the eyes as well? It's something
Yes.
I used to hear
Yes.
about. But
Yes, it does affect the eyes. You can see similar deposits, if you like, that you see in that area in the kidney we call the basement membrane, and the equivalent structure in the eye. So yes.
does it affect the vision?
It can do if it progresses.
And does it stop, like, if the transplant's successful and there's no evidence of the disease present?
Coming back, does it stop? I don't know if there's good data. Do you know on that? I'm not sure. Sally?
People seem to have much less trouble with the eyes than the kidneys, so although the kidney function is deteriorating, often they know that the deposits are there but don't tend to progress.